Did you know that 2015 is an anniversary in cell biology? 350 years ago, Robert Hooke published the first description of what he called a cell. But it wasn't until the mid-19th century that we started to look inside of cells. And in 1898, it was the Italian scientist, Camillo Golgi, that introduced us to the cell structure that is the hero of this story, the Golgi apparatus. We now know that the Golgi is composed of stacks of flattened compartments, kind of like a stack of pita bread. And inside the pita pockets, hundreds of chemical reactions occur as the Golgi makes, modifies, and packages materials for the cell. Everything from the collagen in your connective tissue to the plant cell walls that you eat. And every animal, plant, and fungal cell has a Golgi. In humans, Golgi disruption is associated with diseases like Alzheimer's and Parkinson's, as well as a growing list of less well-known disorders, neurological, muscular, skeletal, developmental, and more. But despite close to 120 years of research, we still don't really understand the basic mechanism behind Golgi function. And this is the question that my thesis is helping to address. If you were to cut a Golgi in half, like in this cartoon, inside you would find a mix of what we call residents and cargo. The residents are the workers that live in the Golgi and carry out the chemical reactions that make and modify the cargo, while the cargo is the product and needs to get out of the Golgi. It can be helpful to think of the Golgi as kind of like a can of mixed nuts. Some of the nuts are like cargo, say pecans. You need to pick through them and send them on their way. But other nuts are like residents, like cashews. In order to do their job, they need to stay in the mix. But how do you quickly and efficiently get the pecans out, but leave the cashews behind? Unfortunately, answering this question is extremely difficult because the Golgi is very small, very active, and very efficient. But the system that I'm using lets me work around a lot of these problems, so I can get at the central question of how the Golgi works. And to do this, I'm mapping the location of the residents and cargo within the Golgi. I've created fluorescently tagged residents that you can see glowing in this plant cell on the top right. I can then see where the residents and cargo are in high-resolution images taken with an electron microscope, like the image on the bottom right. The relationship between the residents and cargo and their organization in the Golgi will give me clues about how the Golgi is making and sorting materials for the cell, so that I can build a hypothesis that helps explain how the Golgi works. And ultimately, this research could have an impact in a huge variety of fields, everything from medicine to biofuels to beer, simply because the Golgi is so important for so many different kinds of organisms. Thank you.